Okay, hello and welcome to this tutorial in renal physiology part 2, where we're going to be talking about the regulation of extracellular fluid, acidification of urine, and bicarbonate secretion as well. So it's going to be a very short tutorial. So we're going to start with the regulation of extracellular fluid. We know that one of the main functions of the kidney is to help in regulation of the amount of fluid that you have in your body. So if there's excess fluid, it's going to be excreted, and if there's a deficit, then some fluid is going to be reabsorbed from the urine that is formed, making the urine more concentrated, and so that does lead to as much um, fluid can be lost through the urine, so that you reabsorb as much as you can. So yeah, so the balance of these fluid compartments in the body uh, can happen in two ways. There are main two ways. There are two main ways. Number one, which is the balance of tonicity, and number two, the balance of volume. So what this means is that uh, during the balance of tonicity, you're basically dealing with the dissolved electrolytes that are in the plasma, or the ECF. So tonicity just refers to the amount of solutes in a particular solution. But then for the balance of volume, you're dealing with the direct amount of fluid that is in that particular fluid compartment. So for that, you're going to use another system as well that is going to deal directly with these uh, fluid compartments. So, but for balance of tonicity, what we're basically dealing with is the hormone that is known as vasopressin, or antidiuretic hormone. That's the one that senses tonicity changes, and then it's secreted. So there are receptors that are responsible for sensing the change in tonicity, and then those who send nerve impulses to the brain to release the hormone vasopressin from the posterior pituitary gland. But for the balance of volume, we're going to be dealing mainly with balancing water movement, and that is also attributed to the fact that water always follows where there is more solute. So if you can control the solute that is moving uh, in a particular compartment, you can control the amount of water that you're going to be sending to that particular compartment. So for that, you're going to be dealing with what is known as the renin angiotensin system. So that's what we're going to be dealing with. All right, so we're going to start with the balance of tonicity. So if you look at these two, by the way, they are very intertwined. Both of them involve dissolved solutes and movement of water here and there because of solutes, but the main controls are what's different. So here you're going to be mainly using vasopressin, but here you're going to be using the renin angiotensin system. So don't really bother knowing balance of tonicity or balance of volume. This one is under which, but just know what the hormone vasopressin does, and then just know what the renin angiotensin system is about as well. All right, so the balance of fluid tonicity in the body is mainly controlled by the posterior pituitary gland, like we already mentioned, through the release of a hormone known as vasopressin, or antidiuretic hormone. So this hormone is stored in the posterior pituitary, and then it senses, it's released when the body senses a change in the tonicity of the body fluid. Okay, so when there is a high amount of tonicity, that means there's a high solute concentration with more of a kg of water. That means high solute concentration, there is uh, there is more solute than there is water in that particular compartment, so the tonicity is very high. So you're going to have to dilute that solution. You're going to have to dilute the tonicity. How do you dilute? You always add water. So antidiuretic hormone, what it does, it adds water to a particular compartment, or uh, dilutes the amount of solute that is dissolved in the blood. So how does it do that? It does that by acting on the collecting ducts of the kidney. Okay, it tells them to absorb more water. Okay, so how does this exactly happen? It acts via what are known as V2 receptors. Okay, what do these V2 receptors? So the V2 receptors are on the cells of the collecting ducts of the kidney, and these V2 receptors, once the vasopressin binds to the V2 receptors, it's going to cause aquaporin 2. Okay, aquaporin 2, we did mention of aquaporins when we're dealing with cellular transport. Uh, Types of transport here and there. We know that water moves through channels that are known as aquaporins. From the word aqua, water, a porine is just a pore or an opening. So aquaporin 2 is a type of transmembrane protein that helps in movement of water into and out of the cell. So these ones are stored in the endosomes of the principal cells of the collecting ducts. The cells that have V2 receptors are known as the principal cells, and they're located in the collecting ducts of the kidney. So they have V2 receptors. Once a vasopressin binds to the V2 receptors, it's going to cause these aquaporins to be released from the endosomes that are inside these collecting tubules. So the endosome is just a vesicle containing a particular protein that can be released at will. So it's going to be released to the membrane of this particular cell, and then the aquaporins will be inserted on that membrane, the luminal membrane, the membrane that is facing the lumen. And then what does that do? The aquaporins is going to allow water to move into those cells, and the water can now move from the cells into the interstitium, and then it can go into the blood as well. So that's basically what happens. So water will move from the urine to the interstitium around the collecting tubules, and then eventually enters the circulation, just as I am from saying. So conversely, if there's a low tonicity, what is going to happen is that it's going to inhibit the secretion of ADH. So all what you're talking about is going to be inhibited. The ADH secretion will be inhibited. That means there will be no insertion of aquaporins into the luminal membranes of the principal cells of the collecting tubules. And then what does that mean? That means no water will be reabsorbed. No water will be reabsorbed from the collecting ducts. And then if you have made the collecting ducts relatively impermeable to water, that means uh, the water that is filtered, okay, is going to be excreted like that. The water that is filtered, because remember the collecting tubules is more like the distal part, just before you get into the the calyces and the pyramids. So if you don't reabsorb the water from there, then uh, you're going to lose that water through urine, of course. So here we were forming concentrated urine because we were removing the water from the filtrate that was in the collecting tubules and bringing it back into the interstitium, which then goes back into the blood. But if there is already there's a low tonicity. Low tonicity means there's a high water content and low solute concentration. So that means the water content is high. So you don't need to absorb a lot more if you already have a lot more. So what that does is that it inhibits the ADH secretion. That means no water will be reabsorbed, and then uh, the filtered water that gets into the collecting ducts will just go directly into the calyces, renal pyramids, and then it will go down the ureter and be excreted through the bladder as well, just like that. And then you form dilute urine because that urine contains a lot of water, so it's going to be forming dilute urine. Okay, so this hormone vasopressin also has other actions. It's known as antidiuretic hormone because it prevents diuresis. Diuresis is just loss of water. So antidiuretic hormone prevents diuresis. That's what it means to be antidiuretic. But there's also a component of it. That's what makes it to be called vasopressin. Vaso from the blood vessels, comes from the blood vessels. So it acts on these blood vessels as a vasoconstrictor. So it's a potent vasoconstrictor through the V1A receptors. So V2 receptors, remember, they're the ones that are on the principal cells of the collecting duct that will cause insertion of aquaporins into the luminal membrane of the principal cells to cause reabsorption of water from the collecting ducts. But for through the V1A receptors, these ones cause vasoconstriction of the blood vessels in response to reduce blood pressure. So, how do you control blood pressure? There are two ways. You can either increase the amount of fluid in the blood vessels so that that fluid exerts pressure on the blood vessel, or you can reduce the lumen. You can reduce the diameter of the blood vessel by vasoconstricting it so that the little fluid that you have in there can be adequate to provide that pressure. So you're like reducing the diameter so that you increase the pressure. So reducing the diameter or increasing the amount of fluid that is in the blood vessels are ways of uh, increasing the blood pressure in response to having a reduced blood pressure. So if the blood pressure is reduced, you increase it by those two ways. So there are factors that control the release of vasopressin. Okay, number one, osmolarity of plasma. This one is what we're from just saying. High tonicity, low tonicity, just from what I'm from explaining about. Now, secondly, we have the ECF volume. ECF volume also plays a role. Extracellular fluid volume or plasma volume, blood volume, the one you're thinking about. If the blood volume is low, of course, you're going to want to increase it. How do you increase it? You add more water to it so that you can increase the volume. So, how do you add water? Secrete vasopressin that prevents the loss of water so that it prevents the loss of water and increases the reabsorption of water. So, reabsorbing water will cause the ECF volume to go back to normal. Then blood pressure as well also plays a role because remember vasopressin is also uh, responsible for causing vasoconstriction. So blood pressure, if you have a low blood pressure, it's going to cause uh, the release of vasopressin. But then if you have a high blood pressure, it's going to do the opposite. It's going to inhibit vasopressin so that you don't add a lot more fluid to the blood, because adding more fluid increases the pressure. But if you already have a high pressure, you don't need that. And also, you're going to inhibit vasoconstriction. That means if you inhibit vasoconstriction, you have vasodilation so that the blood pressure can be reduced. All right, and fourth, we have hyper and hypovolemia. So hyper and hypovolemia simply deals with the amount of blood. So ECF volume talks mainly about all of the volume that is in the extracellular compartment, but for hyper and hypovolemia, we are basically talking about the blood content. So the amount of blood that is in the body. Let's say, so hypovolemia simply means you have low blood low blood volume. This usually happens maybe if you have uh, if you have an accident and you lose a lot of blood. That is what is known as a hypovolemic shock. Your body is not able to compensate or gets very overwhelmed from having a very low amount of blood. So during a case where you have maybe acute hypovolemia, it's going to cause release of vasopressin so that there is more water added the to the body so that it increases the extracellular volume, the blood volume as well, because blood is actually in the extracellular volume. So, uh, yeah, hypovolemia will cause the release of vasopressin, while hypervolemia will cause the inhibition as well. Then there is hypo and hypernatremia. Hypo and hypernatremia is just the concentration of potassium. Sorry, potassium, sodium. Yeah, not potassium. Hypo and hypernatremia, that's related to the concentration of sodium. So you have a lot of sodium, uh, the concentration of sodium, if it's high, that means that's high solute concentration. Hypernatremia, high solute concentration. Remember, high solute concentration means we now have what is known as a high tonicity. High tonicity calls for more water to be added to this particular compartment. So this will cause the release of vasopressin, while hyponatremia will cause the inhibition of vasopressin so that water can be lost, so that now we return back the uh, the solute concentration to normal, or you, you prevent the dilution, further dilution of this particular fluid compartment. And then the other stimuli that we can talk about, which is nausea, pain, surgical stress, or some emotions that also cause vasopressin release. So pain, uh, Pain in some areas of the body will cause release of vasopressin. Surgical stress, when a person is from undergoing surgery, uh, the body is usually uh, releases some stress signals, usually because there is some volume of blood that is lost there, here and there. So that sends signals to release vasopressin so that uh, you can reabsorb some water from the kidneys as well. So another added note here is about caffeine. Those people who like taking coffee, caffeine is usually an antagonist to antidiuretic hormone because alcohol and caffeine that is in coffee, these are what are known as diuretics. So these um, are going to cause excretion of water. So they prevent the release of vasopressin. Water is going to be excreted. Like that's the reason why after taking caffeine or maybe energy drinks, most people realize that they become dehydrated and weak. The weakness is just due to dehydration. Also, similarly, during alcohol. So during intake of alcohol, after taking in alcohol, most people release. Most people realize that they're actually dehydrated and you have to drink a lot of water, especially if you wake up with a hangover or something like that. They advise that you take a lot of water to, you know, help deal with some of those symptoms like weakness. All right. So clinical significance of this particular part, we have diabetes insipidus. Okay, diabetes insipidus can either be central or nephrogenic. So diabetes insipidus is different from diabetes mellitus because diabetes mellitus is just issues with sugar balance, sugar homeostasis, but diabetes insipidus has an issue with water balance. So it can either be central, meaning that coming from the central nervous system, there is an issue with the nerves that cause release of the vasopressin, or it can be nephrogenic. That means the nephrons are not responding. The V2 receptors that we talked about are not responding to the vasopressin that is released. So therefore, you cannot really control your water levels very well. So that's just diabetes insipidus. That's what it's about. Then polyuria is just a condition where you have passing of more than normal urine. So this can be a result of this. So if diabetes insipidus sets in, then you're not able to release vasopressin, or the nephrons are not able to respond to vasopressin. You're going to be releasing a large volume of diluted urine, and that is what we call polyuria. Then there is what is known as polydipsia. Polydipsia is just you taking in a large amount of fluid. This is just accompanied by polyuria because if you're releasing a large amount of urine, then you're losing a lot of water. That means you have to take in a lot of water as well. So that is what is called as polydipsia. All right, then we can talk about the balance of volume. The balance of volume is mainly achieved through the renin-angiotensin system, like we said. So we'll just get right into the renin-angiotensin system. So the renin-angiotensin system uh, basically involves angiotensin acting on either the cardiovascular system or the adrenal cortex producing aldosterone. So it basically involves uh, angiotensinogen being released, being changed to angiotensin 1, then and the angiotensin 1 will be changed to angiotensin 2. Angiotensin 2 is the one that is going to act on the cardiovascular system or the adrenal cortex. So let's just go a little bit into detail through this. So angiotensinogen, whenever you hear angiotensinogen, if you add gene uh, to anything in the body basically that is released or a hormone, that's a precursor. That means it's the inactive form. So the inactive form angiotensinogen is produced in the liver, and then the kidneys in response to low volume, okay, or low blood pressure. Okay, low blood pressure is going to cause the macular denser cells in the kidneys. I'm sure we did that in renal physiology part 1 or anatomy renal physiology. Must have learned about the macular denser cells. They are also baroreceptors. That means they sense the changes in pressure. So if there's low pressure, the macular tensor cells are going to sense that, and then they're going to release renin. Okay, renin is going to be released by those cells in the kidney, and then what the renin is going to do is going to change angiotensinogen into angiotensin 1. Okay, the renin is an enzyme that acts on angiotensinogen produced by the liver to angiotensin 1. Angiotensin 1 has to be converted into its active form, or another conformation which is known as angiotensin 2, that is the one that is going to act. So angiotensin 2, uh, angiotensin 1 is converted to angiotensin 2 in the endothelium. Okay, there is what is known as endothelium of blood vessels, and also the enzyme is also produced in the respiratory system in the lungs. Okay, in pulmonary system. So in the endothelium and also in the respiratory system, you're going to have what is known as angiotensin converting enzyme, and then that converts angiotensin 1 into angiotensin 2. Angiotensin 2 is the one that is going to act on the cardiovascular system. What does it do to the cardiovascular system? It causes vasoconstriction. Then the other action of it is to cause uh, the adrenal cortex, particularly the zona glomerulosa, to release aldosterone. Remember, the zona glomerulosa releases mineralocorticoids. Mineralocorticoids, the most potent one is aldosterone, and aldosterone is going to cause the reabsorption of salt or sodium from the uh, from the kidneys. And then that, you know that if you reabsorb salt or this sodium, we know that if you are increasing the concentration of sodium in a particular area, that causes the increase in tonicity. An increase in tonicity that we are from explaining causes water to be reabsorbed. So water is going to simply follow the sodium. We did this particularly in the very first videos when you're talking about the ECF volume, where we talk about the concentration of water volumes. So we talk about the concentration of water volumes in different compartments due to the amount of solids that is in them. So controlling the movement of sodium in different compartments helps regulate the water movement. Okay, so that's what it does. So remember vasoconstriction, like we said, there are two ways of controlling blood pressure. Like at the beginning here, we said that a renin enzyme is secreted when there's low blood pressure. Okay, low blood pressure, that means there's low volume of blood. Okay, so vasoconstriction, that means the vessels are going to be constricted. Like I said, there are two ways of increasing the blood pressure. You either vasoconstrict it, increase the cardiac output, and also increase the water content in the blood vessels. So here it does two of these: vasoconstriction and uh, increasing the water content by salt retention. So the blood pressure will be increased conversely, like we had said. So master this. Liver produces angiotensinogen. The liver will produce renin in response to low blood pressure. The renin will act on angiotensin, changing it to angiotensin 1. Then angiotensin 1 will be changed to angiotensin 2 by the angiotensin converting enzyme ASCE, that is found either in the endothelium or produced in the respiratory system as well. Then angiotensin 2 is going to act on the cardiovascular system, causing vasoconstriction, and also act on the adrenal cortex by releasing aldosterone. Aldosterone is going to cause reabsorption of salt. If you go back to hormones in the one we did in physiology, we know that aldosterone acts on salt retention, and then that also causes water retention, and then these two will cause uh, uh, increased blood pressure, sending it back to normal if you had a low blood pressure. All right, so that's that uh, about the renin angiotensin system. Maybe some things you can know that we've already mentioned: pressure sensors in the intrarenal region, that means inside the kidney, And the precious sensors, particularly maybe they can be found in the macular denser. You can talk about the macular denser cells. Okay. Then the heart also produces what are known as natriuretic peptides. There is arterionatriuretic peptide, natriuretic peptides. That means these cause excretion of sodium and eventually, so natri from excretion of sodium, uretic from excretion of water. They cause the excretion of water. That's what ANP, BNP, that's what they do. So you hear of arterionatriuretic peptide uh, and beta natriuretic peptide, something like that. All they do is just cause excretion of sodium, and eventually water follows the sodium, so water is excreted as well. So yeah, basically that's that. Then the name, I forgot to tell you about the name angiotensin. Angiotensin comes from, apart from its action in the adrenal cortex, it also has a function in the cardiovascular system, like I said. So angio, remember angiogenesis, uh, that it has to do with blood vessels. So angio, blood vessels, tensin, it causes uh, tensing of the blood vessels. So tensing of the blood vessels is simply the vasoconstriction. So that's where this name comes from. Angiotensin is a vasoconstrictor from its name. All right, we're going to this other part, which is acidification of urine. So acidification of urine, I'm going to go over this quickly the next 2 minutes. It's just how the the kidneys help in maintaining uh, a proper acid balance, a proper pH balance in the body. So it can do this either by secretion of hydrogen ions or retention of these ions. So secretion of hydrogen ions is going to occur if there is a high concentration of these ions. So if there's high concentration of H plus ions, that means there's high acidity in the body, then these have to be excreted. Some of them have to be excreted. Okay. But then if you retain them, that means the body is under alkalosis. That means there is less H plus in this. So remember that the body works under a narrow pH range. That means it has to be controlled. Okay, so it has to be controlled, maintained. It can't go very low, it can't go very high. So it has to be maintained. So this is part of the way in which the renal system compensates for those changes in the pH that you're going to be finding in the kidneys. All right, so secretion of H plus ions, how basically this happen is summarized in these pictures. So remember this reaction, carbonic anhydrous works on carbon dioxide and water to produce CH2O, carbonic acid. This carbonic acid will dissociate into a proton and a so it will dissociate into this proton and this bicarbonate ion. So the proton will be exchanged for sodium on the luminal tubular lumen. So this is where the tube is, and this is where the interstitium is. All right, this is the lumen of the collecting tubules there, somewhere there. So sodium is exchanged for this if you have to excrete it, and then the sodium concentration in here will be exchanged for the potassium. The reason why this happens is that as a lot of sodium is getting in here, you're going to have a build up of sodium in here. So the sodium has to be, it will go down its concentration gradient as well. So as it's going down its concentration gradient, K plus enters the cell as well as more like a regulator, because if you have you're going to reach a point at which you have excreted a good amount of this and you don't want to excrete it anymore. So that coincides with the high concentration of the potassium that would have entered the cell at that particular moment, and potassium will inhibit the secretion of uh, these uh, these ions. So if then you have a high amount of these that need to be excreted again, the potassium can leave passively like that. All right, I hope that makes some sense. So you start this cycle here where carbonic anhydrase, then this weak acid is dissociating into a bicarbonate ion and a hydrogen ion. The hydrogen ion will be exchanged for a sodium, and it will be excreted like that. Yes, and then the sodium will be exchanged for potassium again in the cell. So basically that's what is happening here. While this other bicarbonate ion will go into the interstitium like that. So for every one thing to note here is that uh, for every proton that is transported out of this cell, you are going to have the interstitium gaining one sodium and one bicarbonate ion. So that is what is happening. One sodium is being gained by the interstitial fluid, and one bicarbonate is being gained by the interstitial fluid for every proton that is lost to the tubular lumen. Okay, I hope that is okay. Then there is what is known as fate of the secretion of H plus ions. So if you secrete it like that, there are fates that can happen, or what happens to these hydrogen ions that are secreted. Number one, they can recombine with the bicarbonate that is in the tubular lumen. There's still some bicarbonate in there. They form water, and then the carbon dioxide is returned back so that it can undergo this reaction again. So it's the carbon dioxide can be returned back for the buffer system. Remember that this is known as a buffer system, the carbonic acid buffer system. Then another fate is there's also this buffer system, the dihydrogen uh, phosphate. Dihydrogen phosphate buffer system, the same thing again. This can be used in this buffer system where sodium is involved. Then another one is if you want to excrete it completely, if you want to excrete the hydrogen completely. So this is a way of excreting hydrogen in the sense that you retain the carbon dioxide back. For this one, it helps in retaining the sodium back. But then for this one, you are just excreting it as ammonia. So it's going to combine with an ammonium ion. The hydrogen is going to combine with an ammonium ion and form ammonia, and then that is excreted together with whatever is passing through this. Remember that ammonia is part of the urea cycle, something like that, and then yeah, that is biochemistry basically. But yeah, as a waste product is excreted just like that. So factors affecting hydrogen secretion in the kidneys and bicarbonate excretion as well. So partial pressure of carbon dioxide. If the partial pressure of carbon dioxide is high, that means there is going to be high amounts of acid formed through the bicarbonate buffer system. So that is going to cause, if you increase rate of acid, so high partial pressure means high acid concentration, that means a high excretion of hydrogen ions. The opposite is also true. An inhibition of carbonic anhydrase. If you inhibit carbonic anhydrase, that means there won't be production of the hydrogen ions. So that means you're going to have little hydrogen ions in there. Low pH. Low pH means you don't have to excrete them. So that means you're going to inhibit the secretion of H plus ions and stimulate their reabsorption. K plus concentration in a nutshell just deals with if you have a high concentration of potassium ions, you're going to inhibit the secretion of H plus ions, hydrogen ions. And if you have a low concentration, you're going to stimulate their secretion. Basically, I think I mentioned it above there. Then adrenocorticotropic hormone concentration. So this one causes reabsorption of salts. So remember that reabsorption of salts deals with reabsorption of sodium. So reabsorption of sodium, if you look at here, sodium will be exchanged for this ion, so it increases the, the secretion of hydrogen in that manner. And then bicarbonate secretion occurs in response to the amount of this ion in the ECF. What simply means for you to excrete bicarbonate, you just look at the concentration of the bicarbonate. If it's a lot, then you're going to excrete a lot of it. But if it's little, you're going to retain a lot of it and inhibit its excretion. But that will change a bit when you're talking about renal compensation, which is the last part. Renal compensation in acidosis and alkalosis. So during respiratory acidosis, that means the partial pressure of CO2 is high. So if you have a high CO2 partial pressure, that is going to cause a lot of acid buildup. Okay, because the partial pressure of the partial pressure of CO2 is going to cause a lot of water to react with it, causing uh, the bicarbonate buffer system, and H plus ions are formed. But then when you form a lot of these H plus ions, you create an acidic environment in the body. An acidic environment has to be buffered. So how does the renal system compensate for this? Like we said, it's going to be excreted. It's going to be excreted. The H plus ions are going to be excreted. But since we know that production of H plus ions also produces these bicarbonate ions, and if these are high, that means these oxidatively are quite high as well. But even if the concentration is quite high, this will be reabsorbed. Okay, instead of being excreted, it will be reabsorbed to add alkalinity while the H plus is being excreted, so that you reduce the acidity, and then add alkalinity so that you balance the pH in that manner. So the opposite happens when you have respiratory alkalosis, where the partial pressure of CO2 is low. There is high concentration of this ion because it's alkalosis. So this ion causes alkalinity. So these ions will be excreted while the excretion of H plus is inhibited. And then the reabsorption of this same ion is increased. So you're not going to excrete the hydrogen, but also you stimulate its reabsorption from the kidneys. So this drops the pH back to normal from its alkaline position. Other than that, there are a few questions that we can go through based on what we have just discussed here. So number one, which of the following is the principal buffer in interstitial fluid? Principal buffer in interstitial fluid. You can, you can pause the video and try to attempt this. So the principal buffer is the carbonic acid. Carbonic acid or uh, sorry, this is not the carbonic acid. Carbonic acid is C. This is the carbonic acid. That is the H2CO2. That's the carbonic acid. That's the principal buffer. It's the one that will dissociate into the H plus and uh, the HCO2 minus. All right. Then secondly, the increased alveolar ventilation increases the blood pH because increasing alveolar ventilation increases the pH because it activates neural mechanisms that remove acid from the blood. It makes hemoglobin a stronger acid. It increases the PO2, the partial pressure of the blood, the partial pressure of oxygen in the blood. It decreases the partial pressure of CO2 in the alveoli, and then the increased muscle work of increased breathing generates more CO2. Right, so the correct answer here is supposed to be D. It decreases the partial pressure. So this option started on the wrong line. It was supposed to say the option D was supposed to say it decreases the partial pressure of CO2 in the alveoli. So that's the correct answer. Because increasing alveolar ventilation, that means you are increasing the breathing rate. That is usually in favor of oxygen. So if you increase the amount of oxygen that you're taking in and increase the amount of carbon dioxide that you're taking out, what that means is that the partial pressure of oxygen is going to increase because you're taking in a lot more oxygen. But then, since you're breathing out a lot of carbon dioxide, that is going to reduce the partial pressure of CO2 in the alveoli as well. So that causes respiratory alkalosis because reduced partial pressure of mm, CO2 causes little production of this. So that means this will not be produced in as much content as possible. Then that is going to remember that drop in H plus ion concentration simply means alkalosis. So that is the increase in pH. Remember, increase is towards the alkaline end, and then pH drop is towards the acidic end. So that is the answer there. All right. Other than that, this is the end of this particular tutorial.